Thank you very much. So thank you to the organisers for the opportunity to tell you today about our work looking for new diagnoses in this RNA sequencing data from the 100,000 Genomes Project. It's a very, very new data set, so this is quite preliminary analysis, but we're really thrilled to share this with you today. So the diagnosis of rare disorders has really been revolutionized in recent years through the advent of whole exome and whole genome sequencing. But even in cohorts with whole genome sequencing done, about half of patients don't get a diagnosis. And a lot of this is to do with the fact that our ability to generate this data has really outstripped our ability to interpret it. And that's particularly true for variants that fall in the non-coding regions of the genome or variants that affect splicing. RNA sequencing gives us a way to kind of get around this interpretation bottleneck and see what's actually being produced from the gene at the RNA level. So we can look for places where patients differ from controls or from other patients in terms of expression or splicing, and link that back to changes in their DNA and ultimately make diagnoses for the patients. The 100,000 Genomes Project was initiated in 2012 with the aim of catalysing the use of genomics in healthcare and in research. As part of the project, they ran a diagnostic pipeline where they did variant filtering and applied panels, gene panels relevant to each patient's phenotype through PanelApp. And this came up with a diagnostic rate of about 25%, so there's still big room for improvement there. At the time of recruitment, as well as gathering the whole genome sequencing samples, they also collected various multi-omics samples, and it's these RNA samples that I'll be telling you about today. So the data set that I'm talking about is whole blood-based RNA sequencing, which has had globin depletion and ribosomal RNA depletion done. And at the moment, we have that data on about 5,100 probands. We're actually expecting the last delivery of data later next week, so that's going to increase slightly in the final data set. These are, for the most part, from probands that didn't get a diagnosis from the whole genome sequencing data. So what I'm mainly going to be talking about today is some early work that we've done looking for expression and splicing outliers using these tools that I've highlighted here. And once we've identified places where each proband differs from other probands, we're using them as kind of internal controls. We can apply gene panels and link that up to the phenotype that's observed in the patient, link it up to their whole genome sequencing data, and ultimately make new diagnoses. Oops. So I don't want to dwell too much on QC for this data set, but I will just say that it's really high quality and has a really high depth of coverage. So we've got a median read count of 102 million mapped reads for each proband, and that's really incredible. So I'm not going to be talking about the full 5,000 for most of the rest of the talk. I'll be focusing on 4,438 and subsets of that 4,000 that were available when I put this abstract together and that had at least 60 million sequencing reads. So this slide shows an overview of the demographics of this 4,400 probands. So you can see from the graph that the majority of these probands have neurodevelopmental disorder type phenotypes. And we've also got a good representation of cardiovascular and renal diseases, as well as lots of other phenotype types too. This is broadly representative of the Genomics England data set as a whole. There's not a particular enrichment for any sort of disease class. So one of the questions that we're asked most often is, how well are you going to be able to assess these phenotypes in a data set of whole blood RNA sequencing? It's not the obvious tissue of choice for the majority of these disease disorders. So this plot aims to address that. It's using the metrics transcript per million, or TPM, and this is a way of normalizing and quantifying the level of expression for the genes. There's no kind of hard and fast cutoff for this, but we found in our experience, if a gene has a TPM of about 5, you should be able to assess most splicing events in most people. And for expression analysis, you tend to need a bit lower coverage. So if you can assess splicing, you can generally assess expression. And what we find in this data set when we look genome-wide is that we're reaching this level of a TPM of 5 for about half of all genes. And when we restrict down to the genes that we're most likely to be interested in, this proportion's actually quite a bit higher. So across all of the genes in PanelApp, we should be able to assess about 60% of them. And when we restrict that down to just the most likely disease genes for this cohort, the majority of which have neurodevelopmental disorders, just the intellectual disability panel, about 73% of those genes reach this level of coverage. So we're really positive that we should be able to assess most disease genes using this data. It's not moving on. Oh, now it's moved on a lot. The screen here isn't working, so I have to keep looking there. I don't know if that can get sorted out. Okay, so on to some expression outlier results. So to look for expression outliers, we have used the tool Outrider, which we've run via the DROP pipeline. And this has been able to identify genome-wide about 4,000 total expression outliers across about 1,347 patients. And when we restrict those events down to the PanelApp panels that were applied to those patients, to the panels that are most likely to be relevant to their phenotypes, we're down to about 0.13 per person. And that's about 7.6% of the cohort that have one of these events. So I wanted to give an example of one of these expression outliers. So this is in the gene RPL5. This is a gene that's associated with an autosomal dominant diamond blackfan anemia, which is consistent with the phenotype observed for this patient. So RPL5 was detected as an outlier with a level of expression of about 0.55 versus the controls. So in this patient, they're expressing the gene at about half of the level of the controls. And the eagle-eyed among you will have spotted that in this DNA sequencing trace, there are some real telltale signs of a deletion that's taking out the first half of this RPL5 gene. But it's not just this particular gene that's taken out, it's actually a much larger deletion than this. It's about 900 kb. And there are several different genes in this area. So this gives us a nice way to look what Outrider might be able to detect versus what it might not be able to detect. So these are the Outrider results at the bottom of this table here. So you can see that 4 different genes have been detected as significantly differentially expressed. And these tend to be the genes that have the highest level of coverage. So if the TPM is over 6.5 in this case, it's been able to detect them, with the exception of this GFI1 gene, which we think it hasn't detected because even though it's got a reasonable level of expression, it's a very small gene. So the level of data to kind of assess that properly just isn't there. In terms of splicing outliers, we've used the tool Leafcutter MD, and this is on the full 4,438 probands. And when I initially kind of ran the analysis and did multiple test correction, I ended up with over 700,000 different differential splicing results, which is loads. And as I started kind of looking at these results, I noticed certain patterns that were leading to clusters of false positive results. So these were genes that had particularly low expression, significant events but that had very small effect sizes, and events that were called as outliers in a large proportion of the cohort. So I instigated these filters to kind of address some of these false positive modes and actually got down to quite a reasonable amount, most of which looked like they're genuine splicing events. So per person, that's about 4 events genome-wide, and when we restrict down to just those PanelApp relevant genes for each proband, that's about 0.17, and again, that's about 8% of the cohort that has one of these fairly credible splicing outliers. I was really excited to see the preprint that talks about Fraser-2 come out recently. So this is very preliminary. I've only run it on 200 samples so far, but Fraser-2 has been designed to detect these splicing outliers, but specifically focusing on the ones that are most likely to be biologically relevant and trying to cut down on those false positives that's quite typical of splicing analyses. So restricting down to the disease-relevant genes, that's about 0.2 per person, or about 13% of the cohort that have one of these outliers. It is worth just noticing that the overlap between the events identified by Fraser 2 and by leafcutter is actually really low, and this is something that we've seen in our own analysis, in other data sets, and it's quite widely reported in the literature too. These splicing tools all tend to identify quite different things, and that's why it's really important for us to use several different tools so we can pick up the most amount of stuff. But obviously then that introduces this issue of false positives. So I wanted to give an example of a splicing outlier. So this is in the gene P10, which is causal of Cowden syndrome. And again, this is consistent with the phenotype that's seen in the patient. And this was an event that was identified by leafcutter with a fold change of 0.59. And this basically means that leafcutter is seeing a new splice acceptor site being used in the proband that's not present in the controls, and it's being used in 60% of the sequencing reads in that control. In that patient, sorry. So this is the key area of the sashimi plot in the middle, so you can see that between these two exons in the controls, there's just a single splicing event, while in the proband there are two different splicing events. And if we zoom in to look at the individual sequencing reads, you can see that although the proband does have some splicing happening at the proper splice site where this intron exon boundary is, they also have this kind of shoulder of coverage that extends out into the intron and is associated with this DNA variant here. So this is a single nucleotide change that activates an AG dinucleotide in the DNA sequence, activates a cryptic splice site, and makes that AG look more like a splice site than the proper splice site AG. So that starts getting used instead. And this variant would have been missed by Genomics England's diagnostic pipeline and by most diagnostic pipelines. It's 20 bases away from the exon, so it's just annotated as intronic and would be filtered out. So obviously it's quite early days for this analysis, but I wanted to try and make an estimate of what proportion of the cohort we would expect to find one of these credible outlier events in a disease-relevant gene. And we think, kind of taking all of these results together, that we'll find one for about 25% of the cohort. And as we add in additional tools and analysis methods as well, we would expect that to increase further. So in terms of limitations, I've already mentioned how splicing is tissue-specific and expression's tissue-specific, and obviously blood isn't the ideal tissue of interest for most of these disorder types, but we actually think that we should be able to assess the majority of disease-associated genes using this data. There's obviously lots still to do, and I won't go through this point by point, but I think it is just worth mentioning that as well as kind of this discovery work where we're looking for novel events in these probands, there's also quite a large fraction of the cohort that got a variant of unknown significance from their genome sequencing, and we'll be able to assess these variants of unknown significance in the RNA sequencing data and try and clarify those impacts as well. So just to conclude, I've told you about this new, really exciting RNA sequencing data set from Genomics England with over 5,000 probands that have rare disorders, the majority of which are undiagnosed. And this data set will actually be available for registered Genomics England researchers later this year. We've already been able to identify many expression and splicing outliers, some of which we already think are diagnostic, and the work on this is ongoing. But our early estimates say that we should find one of these candidate diagnostic events in about 25% of the cohort. This has the potential to make a huge impact on diagnostic rate, given that that only stands at about 25% as it is. So thank you all to the people that have made this data set happen. It's been a huge team effort. And thank you all for listening, and I'm very happy to take any questions.
So question for Jenny. We have time for 2 questions.
Hi,
Please.
Jenny. I'm Maria from Germany, German Center for Neurodegenerative Diseases. I was wondering about the diversity of your cohort. Was it one single population or multiple populations?
So it's all from the UK, kind of clinically ascertained during the recruitment period of the 100,000 Genomes Project. So it's pretty representative of the UK population as a whole. From self-reported demographics, it's about 70% white, and then there's kind of a whole mix of other kind of ethnic backgrounds as well. So it's predominantly white British, but there are also diverse elements to it as well.
If you're working with only blood sample, I think getting blood from different populations would be easier for you. You can add from different populations. For example, Africa, I would like to emphasize so that we can be more sure about the data results.
Yeah, absolutely. No, that would be great going forwards.
Okay, good question there.
So, here. So I'm Carla Oliveira from Porto. My question is related with the tissue specificity. So do you consider that finding this in blood is representative of the tissue where the disease may arise?
Yeah, so broadly speaking, blood isn't going to be a sort of ideal tissue of choice for this sort of analysis. Ideally, if you're looking at neurodevelopmental disorders, you want a sample of the brain, but we know that that's not possible. The blood is the most widely available clinically accessible tissue, so if we can make this diagnostic process work in blood, that's going to be the absolute dream, because that's going to be the way we can apply it to most people and get the most diagnoses. And I think that, yeah, from our early data, it looks like we should be able to assess the majority of these variants, even though you wouldn't necessarily think that it was the tissue of choice.
Thank you.
Thank you, Jenny. Our next